Hi, my name is James Trunhill, and I'm a third-year medical student at the University of Nebraska Medical Center in Omaha, Nebraska. This module is about an immunological theory called linked recognition. This topic was chosen because it's a great way to tie together several immunological concepts while linking them to a real-life application, vaccine development. My hope is that you will find value in why we learn these different scientific theories and understand that they really do have value in how we practice clinically. These are the objectives of this module.
Linked recognition, and immunology in general, is a potentially difficult subject for many medical students to conceptualize. In reality, neither is difficult to understand once you get the basics. Remember that antibodies and T-cell receptors recognize epitopes on antigens. Armed with this information, it is easier to understand that linked recognition is the process by which a T-cell receptor and surface immunoglobulin recognize different epitopes on the same antigen, resulting in a strong immune response to the antigen. The concept of linked recognition is important when understanding how vaccines can be developed, which I will further expand at the end of this presentation. Let's now start the basics. We have a cell. This cell happens to be infected by a virus that has three epitopes, which are triangle, circle, and square. As the virus replicates within the cell, it will create different parts of the virus that will eventually assemble to form a new virus. Early on in the process, this will result in multiple copies of the three major epitopes floating in the cytoplasm, as depicted here. Do you remember that major histocompatibility complex thing you learned about in immunology? Well, it's kind of important in linked recognition. Remember, there are two major types of MHC. Class 2 is expressed mostly on antigen-presenting cells, or APCs, such as dendritic cells, B cells, and macrophages, whereas Class 1 is present on all nucleated cells. Class 1 and Class 2 are promiscuous. That is, they put whatever material they can fit into the epitope binding site into the epitope binding site. In this case, MHC class 1, which is found on all nucleated cells in the body, is presenting the three different epitopes for this particular virus. If an individual is lucky, they will have a T cytotoxic cell, or TC cell, wherein CD8 will bind MHC class 1 and the T cell receptor will recognize one of the epitopes. Remember, the T cell receptor on a TC is specific for one epitope, and recognition through this process, with some other co-stimulation, will result in the lysis of the infected cell and release all of its cellular contents into whatever space it lies. Importantly, the TC will replicate into tens of thousands of identical cells in anticipation of other virally infected cells. This will complete the two major tenets of the adaptive immune response, specificity and memory. The lysed contents of the virally infected cell will travel through the lymphatics to the lymph nodes, eventually reaching to the circulatory system and the spleen. In the spleen, the viral epitopes move into the periarterial lymphoid sheath, or PALS. The PALS are composed of 1. A T-cell rich area directly around the arteriole, 2. A germinal center, or B-cell rich area, and 3. An antigen-presenting cell-rich area that sits around the germinal center and between the T and B cells. Like its name suggests, the PALS surrounds the central artery. This arrangement makes the area easily accessible to different epitopes. In the PALS, as the epitopes from the cell lysates percolate out from the arteriole, the epitopes will encounter APCs, such as macrophage in this illustration, bind and be internalized via the pathogen recognition receptors, or PRRS, and process or degrade the epitope. After the macrophage internalizes and processes the antigen, it presents the epitopes in the peptide binding groove on its MHC class 2 molecules that are only expressed on APCs. Since the MHC class 2 molecules are promiscuous, it is possible that any or all of the epitopes, the circle, the square, or the triangle, are going to be presented. This is probably a good time to take pause in the presentation and again reiterate that this is a very complex process. There are tens of thousands of epitopes on each viral cell. We are only showing 3 for the simplicity's sake. MHC class 1 and 2 may present the same epitopes, or they may not. But for simplicity's sake, we're going to assume that MHC class 1 and 2 are presenting the same epitope to make this a little bit easier to understand. Just remember that immunology, like nearly all science, is a theory, and that our understanding of the topic is increasing with time. When the macrophage or APC recognizes the viral antigens through its PRRS, pattern recognition receptors, this results in the activation of the cell-to-release interleukin-1 and the expression of the second messenger or co-stimulatory molecules B7-1 and B7-2. CD4 positive T cells from the T cell rich area of the PALS will interact with the MHC class 2 on the APCs expressing viral epitopes through their specific T cell receptor. The T cell becomes activated and expresses CD28, which reacts with B7-1 and B7-2 co-stimulatory molecules on the macrophages. This signals the CD4 positive T cells to release interleukin-2, upregulate the high-affinity IL-2 receptor, and result in proliferation. Here you can see the TH cells proliferating in the T memory cells specific to the receptor. As the lysate continues to percolate away from the central artery, it will encounter the germinal center, or B-cell-rich area. If the viral epitopes are still present at this point, the surface egg, which is the B-cell receptor, may specifically bind, internalize, and process them. I have to make a side note about surface egg right now. Each surface egg is specific for a specific epitope shape, and its specificity can never change. This is due to VDJ gene recombination, which is an entirely different immunological concept that deserves its own lecture. What you need to remember is that surface egg is an antibody that recognizes a specific epitope on an antigen. It is imperative that one remembers that a major function of the B cells is to also serve as an APC. Therefore, once the B cell internalizes and processes the antigen through the interaction of the antibody to the epitope on the antigen, it presents the epitopes in the peptide binding groove on its MHC class II, just like the macrophage did before. Thus, in this example, the MHC class II molecules on the surface of the B cell have the ability, just like the macrophage, to put all three peptides, the circle, square, and triangle, of this particular pathogen out on the cell surface. Importantly, the epitope of the viral antigen that the surface egg on each individual B cell recognizes does not matter, as all of the epitopes will be presented on MHC class II. The CD4 positive T cells will interact with the peptides expressed via MHC class II with their specific T cell receptor. The T cells are now activated by the macrophage and will release cytokines, especially IL-1 and IL-2. These cytokines will exacerbate T helper cell proliferation until tens of thousands of T helper cells are made, along with T memory cells. Both macrophages and B cells are antigen-presenting cells and will increase the number of T helper cells specific for a unique epitope. The binding of T helper cells to the MHC class II and the secretion of various cytokines will also result in the activation of CD40 ligand on the surface of T helper cells. The interaction of T helper cells with B cells will result in the upregulation of CD40 on B cells, which in turn will interact with CD40 ligand on the T cells to induce the second signal, or co-stimulation. As CD40 and CD40 ligand are expressed, the T helper cells proliferate and are made into T memory cells. These cells will be the big shots the next time the virus is encountered and will help offer long-lasting immunity to the antigens, so the entire process can be sped up in the next encounter. B cells express surface egg through gene rearrangements, and the specificity cannot be changed. So in this example, the surface egg of the B cell recognized the triangle and the whole antigen was bound, internalized, processed, and all three epitopes were presented on MHC class II on the surface. At this point, any T cell that recognizes any three of the epitopes present on MHC class II can help the B cell to become a B memory cell or a plasma cell. The B cells that differentiate into plasma cells through the process secrete antibody with the same specificity as the surface egg which bound the viral antigen. This process can result in 1 T cell being able to activate many different B cells to become B memory and plasma cells, 2 one B cell activating many T cells to become T memory cells, and 3 multiple T cells being able to activate one B cell to become a B memory or plasma cell. The primary response creates a short-lived IgM antibody response. However, with T cell help, the B cell can switch isotype. Plasma cells are short-lived and produce large amounts of antibodies with one specificity, and then they die. They do not express a large amount of surface egg, cannot express MHC class II, and therefore cannot be antigen-presenting cells and cannot class switch. That is, they only work for one specific antibody. Because they create such a large amount of antibody in a short period of time, they help T cells by tagging infectious material so it can be easily identified by T cells. Had different B cells with different specificities encountered the other epitopes on this virus, they would have been made into plasma cells that would make antibodies with different specificities.
If you're like me, you might be wondering, why should I care about this? Let's take a look at this theory in the context of a real-life example. The process of a vaccine causing immunity is very similar to that of a virus or bacteria. You know the concept. This is why we give vaccines. If you're having difficulty with the concept, review the discovery of vaccines by Edward Jenner in the late 1700s. This vaccine has a carrier molecule, typically an antigen of high molecular weight, able to cause an immunological response in vivo. And a hapten. The hapten is an incomplete antigen, usually a carbohydrate, and is not able to make a response on its own. They become immunogenic when coupled to the carrier.
For the conjugate vaccines, the haptens are often parts of an infectious agent to which the immune system cannot efficiently react, such as a polysaccharide. Typically, vaccines contain several different haptens and are so-called multivalent. The particular vaccine shown above is 7-valent, just like Prevnar was when it was first introduced. The vaccine contains salt polysaccharides of the capsular antigen of strep pneumonia, serotypes 4, 6b, 9v, 14, 18c, 19f, and 23f, but that's not super important. The carrier protein to which it is attached is a non-toxic recombinant variant of the diphtheria toxin called CRM197. CRM stands for cross-reactive material. It is known to generate a very strong T helper response in vivo. When the vaccine is injected, the immunogenic carrier peptide will be taken up by an antigen-presenting cell. In this instance, it is taken up by the PRR of a macrophage. The macrophage will internalize the vaccine, process it, and present the peptide from the carrier protein on its MHC class II. Remember, the haptens are just simple carbohydrates and do not have the ability to bind to MHC class II. However, the protein part of the carrier, the diphtheria part, can bind there. Hence, this is the reason why it is a potent Th cell antigen. Activation of the macrophage also results in the release of interleukin-1, which activates more macrophage and T cells. This will result in the expression of co-stimulatory molecules B7-1 and 2. When the macrophage encounters a CD4-positive T helper cell specific for the carrier peptide being presented, the T cell receptor on the T helper cell will bind to the epitope and activate the T helper cell, causing CD28 to be presented and interact with the B7-1 and 2 receptor on the macrophage, resulting in the release of interleukin-2. This will lead to an upregulation of the high-affinity IL-2 receptors and proliferation of the carrier-specific T helper cells to make thousands of these ready to help the B cells. Remember, all of these T helper cells are specific for the carrier peptide. The dose of vaccine administered is large enough that some of it will invariably end up in a B cell-rich area, such as the lymph node or the spleen. A B cell with surface egg specific for one of the 7 carbohydrate units will cross-link the SIG, internalize it, process the molecule, and place the carrier peptide on its MHC class II. In this case, let us suppose the surface egg on this B cell binds to the red carbohydrate and cross-links. After processing the vaccine, the B cell will present the processed carrier peptide in its MHC class II. Remember, these molecules are promiscuous and will present whatever peptide they can fit into their epitope binding sites. At this point, the entire concept of linked recognition becomes very important. Because of the antigen-presenting cell CD4-positive T helper cell interaction earlier, we have an abundance of CD4-positive cells that can interact with this B cell, whose surface egg recognizes the red carbohydrate, but presents the carrier peptide in its MHC class II. CD40 is upregulated on the B cell, and CD40 ligand is upregulated on the T helper cell. This causes the release of various cytokines for class switching, proliferation, and plasma cell production. As I mentioned earlier, there are 3 things that can happen now. 1. The B cell can class switch to become a B memory cell. 2. The B cell can differentiate to become a plasma cell that will in this case secrete red antibody. And 3. The T cell can class switch to become a T memory cell. The memory cells will be helpful for future infections with the viral or bacterial hapten if they occur. The plasma cell response will create a short-lived increase in IgM to the red antigen and fight whatever quote-unquote infection is created by the vaccine. You might find yourself asking, what about the other 6 carbohydrate units on the Prevnar vaccine? If the individual has a B cell with the appropriate surface egg for each of the other carbohydrate units, the same process would be completed. The B cells would eventually present the same carrier peptide on their MHC class IIs for each of the B cells with different antibody specificity. What is important to note for each of these interactions is, no matter what hapten is being recognized by the surface egg on the B cell, the T helper cell specific for the carrier peptide are what create the immune response necessary for linked recognition to work. It is important to remember that the T helper cells are specific to the carrier peptide and not to the hapten. T helper cells to the hapten do not exist. It is the class switching into B memory cells primarily, and also differentiation into plasma cells, that will later allow the haptens to be recognized. Let's bring all of this together. Each of these interactions between the B cell and the hapten specific to it will cause the B cell to 1. Class switch, proliferate, and make B memory cells, and 2. Differentiate into plasma cells and secrete antibodies specific to the carbohydrate unit which the surface egg originally recognized. In theory, we could produce 7 different antibodies using the T helper cell that recognize just the carrier peptide. Also, a large number of T memory cells with the same peptide specificity for the carrier will be created. Thus, One T cell can help many B cells.
We have talked about the theory of linked recognition and how it pertains to vaccines such as Prevnar. Now let's close up this talk by discussing what happens if a person who is vaccinated happens to be infected by one of the strains of strep pneumo against which we vaccinated. Who do we give this vaccine to? The elderly, immunocompromised, people with asplenia, and children who are at high risk of infection and its complications. We don't currently give the vaccine to otherwise healthy people because their immune systems are competent enough to fight the infection, meaning they can create a T-cell response and they are at low risk of pneumonia-related complications. After the immunization was given, we were left with B memory, T memory, and IgM antibodies from plasma cells. The IgM response that occurs to the initial exposure by an infection only lasts for a couple of days, so this does not confer long-lasting immunity. The T memory cells created are specific to the carrier protein in the vaccine. Unfortunately, the actual strep pneumobacteria do not contain this molecule, so B cells cannot express it. This leaves the B memory cells as the workhorses during the post-immunization infection. Strep pneumo is an encapsulated organism. This means that it has a polysaccharide or carbohydrate coat around it to help escape immune surveillance. The B memory cells created are against the epitopes contained within this coat. Here, the strep pneumo happens to contain one epitope, the red triangle. This is a simplification, and in real life it would contain hundreds or thousands of these. The B memory cell in this example is specific to the red triangle. B cells can either undergo T-cell dependent or independent differentiation. T-cell independent differentiation occurs in this case of infection because the B cells cannot express the peptide on MHC class II. The surface egg on the B cell will aggregate around the epitopes on one or several bacteria. This aggregation will activate the B cell. The B cell cannot class switch given these conditions, but it can differentiate into plasma cells and release antibody. The antibody released by the plasma cell will cause the bacteria to agglutinate. This agglutination is attractive for phagocytosis by white cells. In this case, the clump materials are being phagocytized by a macrophage. This process will repeat until the infection is cleared and the patient is healthy.